Dear students, welcome to today's session on regulation of chromatin dynamics, nucleosome assembly and chromosomal organization. Today, we will examine the molecular principles that govern chromatin architecture and its profound influence on gene regulation, genome integrity and disease, covering the following learning objectives: to comprehend and critically analyze chromatin structure and dynamics, to understand the molecular mechanism of nucleosome assembly and disassembly, to assess high-order chromatin architecture and nuclear organization, to integrate chromatin regulation with cellular processes and disease, and to apply advanced experimental and therapeutic approaches in chromatin biology. Dear students, the study of chromatin dynamics, nucleosome assembly and chromosomal organization bridges molecular biology, epigenetics and genomics. Chromatin, a DNA protein complex, is highly dynamic, orchestrating gene expression, replication, repair and cell division. Nucleosomes, the basic repeating units discovered by Roger Korenberg in 1974, transformed our understanding of the genome from naked DNA to a precisely packaged system. The human genome of around 3 billion base pairs must fold into the nucleus while staying transcriptionally accessible. This balance is achieved through nucleosome assembly, guided by histone chaperones during replication and repair, and higher order chromosomal organization into loops, domains and territories that ensure functional compartmentalization and genomic stability. Next, we will talk about chromatin and its dynamic nature. Chromatin, the DNA protein complex of eukaryotic nuclei, compacts the genome while preserving SS for transcription, replication and repair. Far from static, its structure shifts with developmental stage, cellular stage and environmental cues. Proper regulation ensures that genes are expressed only when needed, safeguarding against inappropriate activation or instability. Acting as a molecular switchboard, chromatin dynamics underpins cellular identity, plasticity and genome integrity, making them central to development adaptation and disease prevention. Chromatin states: chromatin, the DNA protein complex in eukaryotic cell, exists along a dynamic continuum between two principal states, the euchromatin and the heterochromatin. Transitions between these states underpin chromatin dynamics, directly regulating gene expression, genome stability and cellular function. Now we will be talking about euchromatin. Euchromatin is loosely packed and transcriptionally active, enriched in gene-rich regions accessible to transcription factors and RNA polymerase. It often carries activating histone marks such as H3K4ME3, which is histone H3, lysine K at position 4 trimethylated, associated with active gene promoters and histone acetylation, which promotes transcription. This open configuration facilitates expression of essential genes such as neurogenic genes during neuronal development and supports processes like cell differentiation and adaptive gene regulation. Next is the heterochromatin. Heterochromatin is densely packed and generally transcriptionally silent, containing repetitive DNA sequences like centromeres and telomeres. It is marked by repressive modification including H3K9Me3 associated with silenced heterochromatin and H3K27Me3 associated with polycom-mediated repression and associated with proteins like heterochromatin protein 1, which is the HP1. This condensed state limits DNA accessibility, maintaining genome integrity, repressing transposable elements, and ensuring proper chromosomal architecture. Now, we will be discussing about chromatin architecture and the fundamental unit nucleosome. The nucleosome, often described as beads on a string, represent the basic repeating unit of chromatin, folding further into a 30 nanometer fiber to enable higher order organization. Each nucleosome comprises of around 147 base pairs of DNA wrapped 1.65 turns around an octamer of core histones, 2 each of H2A, H2B, H3 and H4. Linker DNA, which is 20 to 80 base pairs, connects consecutive nucleosomes, while the linker histone H1 stabilizes DNA entry and exit sites, promoting compaction into high-order fibers. This architecture balances genome packaging with regulatory accessibility. Core histones are small, positively charged proteins enriched in lysine and arginine, facilitating tight DNA interaction. Their N-terminal tails undergo post-translational modifications or the PTMs such as acetylation, methylation and phosphorylation, which modulate chromatin dynamics and gene expression. Non-histone architectural proteins, including the high mobility group or the HMG proteins and the cohesins or the CCC-TC binding factors, also called as the CTCF, further shape chromatin structure. HMG proteins. HMG proteins bind DNA, induce bending and enhance the accessibility for transcriptional regulation and nucleosome remodeling. Next is the CTCF. Cohesin, a ring-shaped complex, cooperates with CTCF, a zinc finger insulator protein, to form a chromatin loops. These loops spatially juxtapose distant enhancers and promoters, orchestrating precise temporal and spatial control of gene expression, genome organization, a long-range chromatin communication. Now, we will see the regulation of chromatin dynamics. Chromatin dynamics refers to remodeling, modification and accessibility changes that regulate gene expression. This is orchestrated in many ways, such as ATP-dependent remodelers. Adenosine triphosphate or the ATP-dependent chromatin remodeling complexes are molecular machines that use ATP energy to alter nucleosome positioning, composition or structure, thereby modulating chromatin accessibility. ATP-dependent chromatin remodelers are multi-subunit protein complexes that harness ATP hydrolysis to reposition, evict or exchange nucleosome, the basic unit of chromatin formed by DNA wrapped around histone proteins. These complexes dynamically alter chromatin structure to facilitate or restrict access to DNA for transcription, replication or repair processes. Next are the switch or sucrose non-fermentable, also called as SWI-SNF. The SWI-SNF complex, named for its discovery in yeast mutants affecting mating type switching and sucrose fermentation, loses DNA histone interactions to enhance gene activation. Second is the imitation switch or the ISWI. The ISWI complex, named for its homology to SWI SNF, promotes precise nucleosome spacing and chromatin compaction, often associated with gene repression. The nucleosome remodeling factor or the NURF and ISWI subcomplex evenly spaces nucleosomes to create condensed chromatin, limiting transcription factor SS in regions like heterochromatin. Third is the chromodomain helicase DNA binding or the CHD. The CHD family, characterized by chromodomains that bind methylated histones, include complexes like CHD1 and MI2, which are involved in transcriptional regulation. The fourth one is the INO80, which is the inositol requiring 80. The INO80 complex, named for its role in inositol metabolism in yeast, specializes in histone variant exchange and DNA repair. It swaps histone H2A Z for H2A, promoting open chromatin for repair processes like double-strand break repair and AIDS replication fork progression. INO80's role is critical in maintaining genomic integrity under stress. The fifth one is the signal responsive dynamics. Chromatin remodelers respond to cellular signals such as phosphorylation, which recruits complexes like SWI, SNF during stress responses, for example, DNA damage. This signal-driven activity ensures rapid chromatin reconfiguration, allowing cells to adapt to environmental or developmental cues. The sixth one is the functional implication of ATP-dependent remodelers. ATP-dependent remodelers are pivotal in gene regulation, development, and disease. ATP-dependent chromatin remodeling complexes are central to the regulation of gene expression and genome stability. By repositioning, ejecting, or replacing nucleosomes, these complexes provide dynamic access to DNA for transcription, replication, and repair. Understanding their mechanisms not only deepens our knowledge of fundamental biology, but also offers insights into human disease such as cancers and genetic disorders. Now, we will see the histone modifications and the histone code. Post-translational modifications or the PTMs of histones are central to chromatin dynamics. They include, the first is acetylation. Acetylation of lysine residues neutralizes positive charges, loosening DNA histone interactions and typically activating transcription. Next is the methylation. Methylation occurs on lysine or arginine residues with effects depending on the site marks like H3K4Me3 promote transcription, while H3K9Me3 and H3K27Me3 repress genes. Next is the phosphorylation. Phosphorylation of serine or threonine residues regulates chromatin condensation during mitosis, DNA damage, repair, and signal transduction, highlighting the complexity of epigenetic regulation. Next is the ubiquitination. It influences both activation and repression depending on the residue and histone involved. These modifications form the histone code, a combinatorial platform that dictates chromatin function. Now, we will see the non-coding RNAs and the higher order regulation. Non-coding RNAs or the ncRNAs are RNA molecules that do not encode proteins but play critical roles in regulating chromatin structure and gene expression, influencing processes like development, genome stability, and disease. By recruiting modifiers or interacting with chromatin, ncRNAs shape gene expression and cellular identity, making them vital for UG studies in molecular biology and epigenetics. Next is the long non-coding RNAs. RNA molecule longer than 200 nucleotides orchestrate chromatin modification by recruiting complexes like polycomb repressive complex 2 or the PRC2. Second are the microRNAs or the miRNAs. Small RNAs around 22 nucleotides indirectly regulate chromatins by targeting the mRNAs of histone modifiers or transcription factors. Now, we will see the nucleosome assembly and disassembly. Nucleosome assembly is tightly regulated process ensuring DNA packaging post-replication or repair. It involves histone chaperones depositing histones into DNA, preventing the aggregation and ensuring proper spacing. De novo assembly during replication. During the S phase of the cell cycle, parental nucleosomes are disrupted at advancing replication fork and subsequently redistributed to daughter DNA strands with the help of proliferating cell nuclear antigen or the pCNA and chromatin assembly factor 1 or the CAF1. CAF1, a trimeric complex, binds newly synthesized histone H3-H4 dimers marked by acetylation at histone H4, lysine 5 and 12, H4K5 or 12AC and deposits them behind the fork. Anti-silencing function 1 or the ASF1 acts as a histone chaperone transferring H3-H4 to CAF1, while nucleosome assembly protein 1 or the NAP1 or facilitates chromatin transcription or the FACT complex assist in assembling H2A, H2B dimers. Spacing of nucleosomes is maintained by remodelers such as remodels the structure of chromatin RCS complex, which positions nucleosomes approximately 165 base pairs apart. Next is the replication independent assembly. Outside the synthesis or the S phase, replication independent or the RI assembly restores nucleosome displaced during transcription or DNA damage. Histone chaperones such as histone regulator A or the HIRA deposits the H3 histone variant at actively transcribed genes, thereby maintaining chromatin openness and transcriptional activity. Histone chaperones and variants. Histone chaperones and variants play a crucial role in maintaining chromatin organization, stability, and proper gene regulation. These chaperones prevent non-specific histone DNA interactions and facilitate the correct deposition of histones during replication, transcription, and repair. Next is the anti-silencing function 1 or the ASF1, which handles the H3-H4 dimers, while nucleosome assembly protein 1 or the NAP1 manages H2A, H2B dimers. Chromatin assembly factor 1 or the CAF1 deposits H3 or H4 during DNA replication, and histone regulator A or the HIRA incorporates the histone variants, which is the H33, in replication independent manner, maintaining transcriptionally active regions. Variants such as centromere protein A or the CENPA replace canonical H3 at centromeres, where holiday junction recognition protein or HJURP assembles them, ensuring kinetochore formation and faithful chromosome segregation. Together, these systems maintain genome integrity and regulate chromatin dynamics. Next, we will see nucleosome disassembly. Disassembly is equally important, particularly during transcriptional elongation and DNA repair. Involves the temporary removal of nucleosomes DNA histone complexes by ATP-dependent remodelers and histone chaperones. Remodelers like switch sucrose non-fermentable and INO80 evict or slide nucleosomes, allowing RNA polymerase II to transcribe genes to repair enzymes to assess damaged DNA, as in double-strand break repairs. Chaperones like facilitates chromatin transcription or the FACT and anti-silencin function 1 or the ASF1 manage histone eviction and storage, preventing loss during processes like replication fork progression. For example, FACT disassembles H2A, H2B dimers during transcription, ensuring smooth polymerase movement. Disassembly is tightly regulated by signals like histone phosphorylation, which recruits remodelers during stress responses. Dysregulation, such as excessive disassembly, can lead to genomic instability, as seen in cancers where SWI, SNF mutation disrupt normal chromatin access. Dear students, we will now discuss about chromosomal organization. Chromosomal organization involves high-order folding beyond nucleosomes, creating loops, domains and territories for functional compartmentalization. This includes higher order chromatin foldings. The 30 nanometer fiber forms loops which are around 50 to 100 kilobase pairs anchored by CCC-TC binding factor and cohesin. HIC maps identify topologically associating domains or the TADs, which are around 100 kilobase pair to 1 megabase pair. Self-interacting regions that insulate enhancers, promoters with lamina-associated domains or the LADs. Tethering to nuclear lamina for repression. Next is the nuclear architecture. Chromosomes occupy territories with gene-rich chromosomes central, gene-poor peripheral. Nuclear pores facilitate transport. Nucleoli organize rDNA. Next is the regulation of organization. The extrusion model involves cohesin looping DNA until blocked by CTCF, condenses compact mitotic chromosomes. Epigenetic marks like H3K9ME3 HP1 promote heterochromatin with Hox gene TAD reorganization, driving developmental expression. From nucleosome to chromosome. Nucleosomes fold into around 30 nanometer fibers, looping and attaching to scaffold proteins for higher order organization. Chromosomes occupy discrete nuclear territories, preventing entanglement and ensuring functional compartmentalization. Next are the chromosome territories. Chromosome resides in distinct territories with gene-rich chromosomes central and gene-poor peripheral. Active genes loop to transcription factories, while nuclear pores and nucleoli organizing rDNA facilitate transport and ribosome biogenesis. Now, we will see typologically associated domains, or the TADs. TADs are self-interacting chromatin domains that act as regulatory units. They help insulate enhancers and promoters, ensuring proper gene regulation. Disruption of TAD boundaries often lead to diseases such as cancer and congenital disorders. The architectural proteins, cohesins, condensins, and CTCF are central in shaping higher order chromatin structures. First, the cohesins. Cohesins hold the sister chromatids together and regulates loop. Next, the condensins. Condensins drive mitotic chromosomal condensation. Next, the CTCF. They function as an insulator protein defining TAD boundaries. Dear students, we will now see the regulation of chromatin dynamics in cellular processes. The first is the gene expression. Chromatin accessibility determines whether transcription factors can bind DNA. Open chromatin, or the euchromatin, favors expression, while the closed chromatin, or the heterochromatin, represses it. Next is the DNA replication. Origins of replication are often located in open chromatin regions. Proper regulation of chromatin ensures timely and faithful replication. Next is the DNA repair. Chromatin must relax to allow repair machinery to assess DNA breaks. Modifications like H2AX phosphorylation signal the recruitment of repair proteins. Next is the cell division. During mitosis, chromatin undergoes dramatic condensation regulated by histone phosphorylation and condensin complexes. Errors in this process can result in aneuploidy. Next is the chromatin dysregulation and disease. First is the cancer. Many cancers are associated with mutations in chromatin regulators such as SWI, SNF subunits, histone methyltransferases, and acetyltransferases. Aberrant histone modification and chromatin misfolding drive oncogenic gene expression. Next are the developmental disorders. Diseases like Rett syndrome, mutations in MECP2 and ICF syndrome, which is the immunodeficiency centromeric instability facial anomalies syndrome, arise from defective chromatin regulation. Next are the neurological disorders. Chromatin misregulation is increasingly implicated in neurodegenerative diseases and psychiatric disorders, reflecting the importance of epigenetic control in brain plasticity. We will now see the experimental approaches to study chromatin. Chromatin immunoprecipitation, or the CHIP. They are used to map protein DNA interactions and histone modifications. This technique links specific proteins or epigenetic marks to regulatory elements, helping understand gene expression control. Next is the assay for transposase accessible chromatin using sequencing, ATAC sequencing, and deoxyribonuclease sequencing, which is the DNA-seq. These methods identify regions of open chromatin, providing insights into regulatory landscapes by mapping accessible DNA. They pinpoint active promoters, enhancers, and potential transcription factors binding sites. High throughput chromosomes conformation capture, which is the HIC, and chromosome conformation capture, or the 3C technologies. They capture three-dimensional chromatin interactions, revealing typologically associating domains or the TADs and chromosome folding patterns. These methods uncover how long-range chromatin looping organizes the genome and influences gene regulation. Next are the imaging approaches. Super resolution microscopy or the SRM and the live cell imaging visualize chromatin dynamics in real time. They allow direct observation of chromatin compaction, movement and structural changes during cellular processes. Dear students, as we wrap up this in-depth theory class on chromatin dynamics, nucleosome assembly and chromosomal organization, you have gained critical insights into the epigenetic mechanism driving cellular function and disease. A deeper understanding of these processes not only enriches molecular biology but also opens pathways for therapeutic interventions in cancer development and neurological disorders. Dear students, carry forward this knowledge to explore innovation, research and therapeutic strategies shaping the future of molecular biology and medicine. This was all about today's lecture. I will see you next time. Thank you.